Welcome everyone to the fifth symposium. I'll try to moderate with Jenny and Barbara's help. We have a lot to get through this afternoon, this morning, not evening for once. 5 lightning talks, 2 keynote speakers, of course, 3 breakout sessions where I think a lot of the discussion will happen, and then plenty of time at the end for Q&A. So before we get started, a little bit of housekeeping from Jenny, fairly standard stuff, I think. Please stay muted if you're not talking. Minimize speaking and barking if you have dogs. Please feel free to use the chat, which we will try to get to and answer all questions as they come up during the Q&A section. Please be patient with us as we go through any technical issues, internet related or otherwise. The video recording will be placed on the Reclone website, so please feel free to join the Reclone network at reclone.org/join, which we'll just post in a few moments. For those who are giving lightning talks, I've been told to be ruthless and give you a 1-minute notice, which is a little unkind, but apparently it's the only way to keep everyone on time. So with that, I welcome everyone. I am grateful for Jenny and Barbara and the team and everyone speaking. And the first agenda item is a talk from India. Shirshendu wears many hats. He is the mission director for Grand Challenges in India, which is a combination of the Ministry of Science and Technology, the Kahlil and the Melinda Gates Foundation and the Wellcome Trust. It plans to support initiatives to dramatically change health outcomes in India and beyond. He brings 25 years of experience across academia and industry and really supports the innovation ecosystem in India. He has many hats, too many to list, covering IP, technology transfer, communications, biotech startup, and connections to tropical medicine. He has a PhD in microbiology, which is laudable, a law degree, less so maybe, and several other business and leadership skills to bring to the table. So without further ado, I welcome him, and we very much look forward to hearing about supporting diagnostic innovation to scale the Indian experience. Please take it away.
Thank you, Keith, for this introduction, and Jennifer and Barbara for the invite to this early morning symposium. And I'm delighted to be here. I bring to be there in the symposium. Bring greetings to all of you from India. A very good namaskar and good morning, good afternoon to all of people attending today. So what I'll do is in next 15 minutes, which has been allotted to me, I will share you the Indian experience of how supporting diagnostic innovation to scale, what is our experience. And I will be giving out a funder's perspective, basically working in a funding agency where we looked at the challenges in funding innovations and how innovations were nearly catalyzed in the academic setting and then scaled up and validated and brought into a commercial exit. That the experience how we do being the funding agency. And very nicely done today. My next speaker today, Dr. Minal, comes from my labs, which has been a really very good live experience, how they scaled up the COVID diagnostic experience. So that I think has been planned very well today. So what I will do is I'll talk about funding agency.
Thank you, sir, for all your support to my lab.
Thank you very much. Yeah, we will discuss that in the question answer session as well. So yeah, I think very well covered today on. So what I'll talk is of the funding agency's perspective. Barbara, can I have my next slide, please? So my next slide shares, what basically the challenges in the innovation ecosystem which we face and how we take it. In case of diagnostics, moving from discovery to delivery is a very quick path as compared to, if you say, vaccines or drugs. In case of medical technologies and diagnostics, it's a very quick path, quick in the sense we can move very, very fast, but there we require strong validation and justification and moving from one step to the other. So we have protocols, and in funding agency, we fund everything from discovery, the proof of concept, moving the proof of concept to a development, more validation phase, and then the delivery phase. So that and the challenges our innovators face at the each level, which is at the discovery, the development, or the delivery, has been outlined in these slides. And this is from our experience and our working, how we face, how it moves around the thing. So when all we advise our young innovators and startups, and my lab is a very live examples of it, is you need to understand the problem well, what you are working for, what is your end product, what is your target product profile, has to be thought in very beginning, because ultimately when the product is formed, somebody needs to buy it. And in the COVID times, it was brought in by the government. So I think how quickly it can detect, how specific is this test, how sensitive is this test, I think that our innovators do a very good SWOT analysis at the very beginning. So we really encourage them to have those analysis done, and understanding the market pull and push, how risky the program is, how much the government support is giving to you, or how much the venture funds are supporting you. I think these sort of analysis needs to be done in the very beginning. And what is the local infrastructure? Though we have created right now a network of incubators in this country, we have created a network of almost 60 plus incubators through our system in this country. But I think everything needs to be analyzed and thought through well in the beginning. I advise all my grantees who come to me that you have to tell us a very good story. How you take your science strong, it has to be 60 to 70% is strong science, and how you convert that strong science into a product which is targeted, which has a high specificity, which has a good sensitivity, which is affordable, which is accessible. I think these all needs to be built in when you think about your story. It should be just like your fairy tale. It should start like once upon a time and ends with happily lived ever after, which is generally a process. So I think that sort of story needs to be developed, and we advise all our innovators working in this space to take it, because this is a very quick moving field. In a 1.5 to 2 years, you know how your product is moving, it needs to be validated, and then the regulatory clearances needs to be brought in. Similarly, when you develop the product, you take it to the development, and then to the delivery, you need to identify all your challenges in place. So that's how we advise our applicants to move forward when they look at diagnostics or medical devices to the next stage. Can I have my next slide, please? So my next slide will tell you where we come from. We come from an organization called the BIRAC, which is the Biotechnology Industry Research Assistance Council. And this has been created by the Government of India through the Ministry of Science and Technology and the Department of Biotechnology to really handhold and enable the innovation ecosystem in this country. That is a very good example of how innovation and BIRAC really handholds innovators, or not only handholds, but enables innovation to take it from bench to the bedside. That is our idea. And our major job is funding. But apart from funding, we enable the ecosystem. We help them to identify mentors. We help them to identify, space or startup incubators. We help them to write their grant application. Once the prototype has been developed, we help them to provide them validation beds to do it. Once they are validated, we help them to navigate through the regulatory system. And once your tests is validated, what the regulatory clearances has been given, and we help them to really connect them to the ecosystem so that it can be bought and sold off. So entire enabling the ecosystem has to be built into a system. So funding is a key element, I fully understand. But if the science is not strong along with the funding and the team is not good, I think you have a number of stories around India, and the next story you will hear after me, the My Lab, is one of such stories where, you have to have a team, strong science, of course, funding, and the enabling ecosystem, which will help you to take your innovation to next level. So my organization really helps to enable the innovation ecosystem, of course, through a funding support, but providing you all the, bricks and mortar for take your innovations to the next level. My next slide, please. So how we enable the ecosystem is we really help them product move from the proof of concept. I will not describe you our programs, but we have programs and bricks from literally from the discovery stage to early and late stage to then to take it to a level of large scale up and then product commercialization. We have programs that really move from all the scales and we really handhold them to take it to the next level. Next slide, please. So this is how we help you to ideate your concept, and our COVID story is the biggest example where in 3 to 4 months, less than 3 to 4 months, good diagnostics were made available. We were initially importing, now we are exporting not only diagnostics but other elements, and this has happened with the strong innovation ecosystem which has been created in India, so that things moves very, very seamlessly from one level to the next level. So we help in ideation, then idea development, proof of concept, early stage development, late stage development, and product commercialization, and hitting you to the market. So you have to handhold the innovation ecosystem so that your innovation literally moves from the bench to the bedside, or from concept to reality. And we have various instruments and mechanisms to support that. Next slide, please. My next slide will tell you what is the key elements in empowering the enterprise. So you need an enterprise structure to take the innovation level, and you should have a space for it to really, breed or visualize your innovation. So we provide incubation space there, and this is government systems, not only us but various other systems also are providing incubation space. Then research resources, academia industry partnership. There are very, very strong academic groups in India, and we help them to catalyze those partnerships between academia and the research to take your innovation from, literally from the research shelves so that it can be validated at a level which can be commercially exploited. IP and technology management, the IP validation and the technology management and mentoring. So all the bricks are needed, which is provided to the ecosystem to take the innovation from literally bench to the bedside. Next slide, please. Now, let us now be specific on our diagnostics, how we really help. And COVID has been a very, very interesting time of supporting diagnostics, because as soon as 30th January, when we first tried to sequence the Wuhan strain, and then the diagnostics companies like my lab really jumped into foray. They were the first to come out with the diagnostic testing kits. So I think that was the time when we also brought out our COVID Research Consortium program, or the COVID Research Consortium grant, which had 4 parallels. Basically, first was the diagnostic, then was the enabling devices like the PPE kits, the thermal sensors, the sanitizers and things. And then we thought, and then the other parallel was therapeutics and the vaccines. You have heard the vaccine story, which I think in a year's time, it was really a history where vaccine really came out from, really the sequence coming out, the virus being isolated and the vaccine being tested. So without bypassing any regulations, that story came out. But the diagnostic was the key element, because without diagnosis, we were not able to move. And this research consortium really, we funded around 19 portfolios were funded. And if you can see, we have a portfolio of, from, under validations to validation, which has been completed in market and scale up. And my lab, you can see in the top right hand in the topmost corner is there. So we really created large scale shared manufacturing facility. One of the AMTZ's platforms, another thing was created, because if you don't get the shared manufacturing facility, quick scale up was not possible. So that was created. Then promoting import substitution. There was a challenge in importing. Flights were closed and a lot of integrants were, coming from various other countries were imported. So we really helped them to, and how we did that, I'll tell you. And then testing and validation support and regulatory facilitation. Regulatory facilitation, testing and validation support was key element to provide test beds. That was created. We have almost now, through our national biopharma mission, which I will briefly describe to you, we have created almost 50 plus test bed sites where you can validate your you have a cohort of positive samples where you can validate it. So we are trying to create that, and regulatory facilitation was a very key element. We opened our doors every week, and every Friday we helped the diagnostic companies to come to us, connected them to the regulators, helped them to how to get them tested, because the startups didn't have that infrastructure to do it. We helped them to connect with other infrastructures to support it. So I think that these enabling platforms were really important to take the innovations to the next level. My next slide, please. So the next slide, what we did was we created NBRIC, the National Biomedical Resource Indigenized Consortium. It was created at a facility in C Camp in Bangalore, where we promoted the idea of Make in India initiative for biomedical research and innovative products, and where we really got them the raw materials and for diagnostics together, and they tried to indigenize them and bring it out. So almost 300 plus Indian manufacturers from diagnostics, from medical devices joined in that consortium, where pool imports of raw material was done, and we helped them to get them these raw materials. Indigenization of raw materials, how can we do it, what can be replaced, a thorough infrastructure, and the government systems was part of this NBRIC facility, the regulators were part of it, the manufacturers were part of it. So you can have a platform of discussion to see how we can take the innovation. And now we have 100, I'm proud to say that companies like MyLab and all other companies, 100% self-sufficiency in indigenous diagnostic production was achieved through this. So I think this was a very good platform which was created, and through the government support to indigenize the diagnostics and become self-sufficient and export it as well. So we are right now exporting it as well. My next slide, please. Few of the products which we have done, if you can see, so many tests have been brought out. So I will not read of them, but this is the example how we really took the brought ourselves to take really a test which was developed post January and was tested, say, around June, July, August. So quickly it was moved to the scale, and not only the innovators needs a full credit of it, which they are the people who did it 24/7, no time limits and nothing, but the enabling infrastructure like the regulators, the validator, people who validated it, and the necessary infrastructure which was created, I think really helped them to take it to the next level. May I have a next slide, please? Now, what I described to you is the DBT, the Department of Biotechnology, BIRAC, and NBMA COVID Research Consortium was created. And under this, these four parallels, nucleic acid detection, antibody detection, antigen detection, and these parallels were really supported under each of these categories were supported. And this was not because I'm telling you that we supported. This was credit to these companies which are named here. They came forward with their innovations, and all were brilliant innovations, which I think had some, because innovations, if they don't have strong scientific rationales, can't be taken to the next level. So I think this was what was achieved, and these are young innovators literally worked throughout and took this innovation to the top. So I think that was really important. I will answer the questions in the discussion forum. Also, I'll try to answer them during my once my talk gets over. Next slide, please. So I will not talk about this. So we supported a lot of screening devices, sanitization platforms, ventilators, and they were all the similar model which was aborted to take it forward to the next level. So I think that was the very important element of supporting across the consortium and creating verticals, which was a diagnostic team was created within our system as well, who were 24/7 talking to these innovators, helping them. We reviewed the proposals overnight. They were reviewed overnight. They were, again, modified overnight. I think both ways, the innovators, our organizations work together in tandem to see that the innovations needs to be moved forward. Whatever funding need was there, we tried to satisfy them to take it to the next level. So next slide, please. So moving ahead, I think what was the learning here was that it was a targeted funding we did. on a specific funding, or we call it hunt and fund. We really looked at proposals, brought them and funded them. So I think targeted funding was a very important criteria. Academia industry translational centers, which really worked out to take it. Partnerships were we didn't bypass any regulatory standard. It was a world-class regulatory standard which was brought in. Scale up and commercialization of innovation was very fast. Robust market pathway. As soon as it was moved, the companies were really had taken up to market very soon, and disruptive technologies was brought in. I think thanks to our innovating culture here, who took it to the next level. And some of more examples. Next slide, please. We also worked on creating mobile diagnostics vans. We have created almost 3 to 4 under the Grand Challenges India scheme, which my colleague run here, Arshi Mahboob runs it, and she, they funded around 3 of diagnostic vans, one in the northeast part, one in the southern end part, one in the 2 in the southern, one in Madras. We created locations. And similarly, this iLab was funded near Delhi somewhere. So these were fully equipped diagnostic vans with RT-PCR kits, so that it can go to the remotest locations, take a sample, get the analysis done, and they were connected through a Wi-Fi network through the central base hospital, where the results were immediately transferred there, and the patients were informed through SMS system, and they can, if they need to be further taken up for further or need to be monitored or quarantined, was done. And similarly, we created these networks of mobile diagnostics labs also, which moved around the remotest rural villages and things, and they collected the data from samples from there, get it analyzed within the mobile diagnostic van itself, and they took it to the next level. Next slide, please, which it shows one more example of it. So this is one which is in the Trivandrum, deployed at Rajiv Gandhi Center in Trivandrum. Kerala was a very hot spot, so we got it deployed there. Next slide, please. Which the next slide shows the internal. These diagnostic van have fully, from disposal of bio waste to doing all analysis was inbuilt inside it, inside the van itself. Next slide, please. So I think basically what it was that we my lab, my colleague from my lab will speak after me, but the first indigenous kit was developed there. Andhra Pradesh MedTech Zone in Visakhapatnam create a shared facility where almost huge number of RT-PCR kits, DNA extraction, RNA extraction kits were manufactured. There were other companies who really supported us in doing it. So I think the enabling funding and the partnership was a key element to take a diagnostics very quickly from discovery to validation to a marketplace. Next slide, please. And this was done through our national biopharma mission, where we really helped them to develop specific products. We create center of excellence to really scale it up, skill development, and technology transfer capabilities from academia to the lab was very key element, which the biopharma mission does it. Now, my final slide talks about what was the key element each should think about when you create these enterprise innovation culture in taking innovation to the next level. We all have to think in a 360-degree approach, where science has to be strong, but apart from science, our exit strategy, our business plan, our IP strategy, our regulatory strategy, a clinical development plan, if it is needed, needs to be built in together to build a strong story to take innovation. And COVID has taught us that. And the infrastructure which we had created really helped us. And COVID has taught us that, where there was a beautiful collaboration between academia, the business, and regulators, the funders, and take it to the next level. So I think that's what we learned. And thank you very much for patience. I think my next slide is the last. This is my last slide. Next slide. Thank you very much. I'm happy to take questions at the end of the presentation from Dr. Minal. Thank you.
Well, thank you for a fantastic talk. I mean, the word that struck out to me was holistic and integrated. I'm sure we'll come back to those questions or observations. Thank you for those who've posted some questions already. Please continue to post questions and we'll follow them through. And Dr. Melanie, you have a great setup You were mentioned several times in that first talk, so the floor is very much yours. And so without further ado, we would love to hear from you about your experience of developing diagnostics. Thank you. Oh, wait, sorry, my apologies. I need to do an introduction.
Hello everyone. First of all, I would like to thank
I need to do an introduction, introduce you, so your background and so on. So Minal has more than a decade of experience in molecular biology and its application to particularly infectious diseases, oncology, and applied science in general. A virologist by training from the National Institute of Virology and with a PhD from California Public University. She previously worked on viral genomics, diagnostics, and particular quality control systems while at NIV. She was instrumental in developing the first ever RT-qPCR test for COVID-19 in India and provided plenty of technical expertise to the MyLab team, particularly with respect to quality control, document management, and commercial the regulatory approvals. Her areas of expertise include developing molecular diagnostics, R&D management, lab operations, and also, of course, training, so important. And she acts as the ISO 17025 internal coordinator. So with that as a belated introduction, her title of the talk is Innovating for a Billion People in the Developing Nation. Dr. Minal, thank you. Minal, are you there?
She has got frozen, I think. Her line has.
Is everybody else there? I guess so. It's one person.
Keith, I can hear you.
Okay, all right. I wondered if it was the Philippines. Thank you again for a fantastic talk earlier on. So much to learn, as Jenny said.
Pleasure. Happy to share. Thank you. Thank you, Jenny.
I'm sorry, there was a little bit issue.
There was
a network issue. I'm really sorry for that. So with the continuation, I would like to emphasize on how India, together with the government and private firms, have fight against the COVID-19 and such a bad disaster. So the biotechnology firm involved in the development and commercialization of the molecular diagnostic kits. And now, starting from the COVID, we have been involved in the serology as well as immunology development kits as well. So today's my topic is innovating for a billion people. So basically what MyLab aim, what is the MyLab vision for starting up this organization is discovery to delivery. And I would like to show our journey in brief. Am I audible? There is a little bit problem at my level about network, so I'm really sorry for that. So basically, we have started a MyLab with a
data. I didn't get it.
Hello.
I have submitted yesterday. They have basically I have submitted some products, which is RT enzyme, tag polymerase, and tag DNA. These are four products which I have registered yesterday.
Can we try and have the phones on mute, please? Thank you.
Should I continue my talk?
For registration, it was pending, right?
Barbara, could you mute? You have host capabilities, I think, to mute people.
Hello, should I continue my talk?
Okay,
Bear with us
thank
1 minute, Minal. I think Barbara is just going to mute any participants that are not on mute, and then you can carry on. Okay, I think we're good. Sorry about that. Over to you.
Okay. We have started with the vision of developing and commercializing the diagnostic kits. We have developed this vision since the swine flu pandemic. I was a part of the National Institute of Virology since 2009 till 2014, where I have been working on swine flu pandemic, its diagnosis and related research. And since then, when I came up with our management team at my lab, we felt that we have a common vision to work for the public health, to work for the society and give the indigenous solutions. Because during the pandemic of swine flu, we were totally dependent on the international solutions. There were a very few companies who have developed the diagnostic test for H1N1. So with these learnings, we have kept a vision for my lab and for India to develop an indigenous test and to support our society. So in 2014, we have started my lab at Pune headquarters, and I was the first employee being at the R&D department. So at R&D, we have started working on the vision, and we have a clear clarity what we wanted to do. So we wanted to start up with the RT-PCR test, which we believe that a gold standard. So we have started working on a NAT testing. First of all, that is a test for blood during for HIV, HBV, and HCV, which is required before donating a blood to anyone. So we have started with this test kit. During whole these years, we have developed an expertise for molecular test kit. It is not a very easy path for us. It is there were so many challenges because at 2014 there was no other firm in India who has developed RT-PCR test kit indigenously. So we were the first one and very independent, no backup, no funding, nothing. But we have started with our vision and with a big dream for society support. So during whole this year, we have developed expertise in the R&D development, where for any diagnostic kit, we need to think about the design of the test kit, its strategy, then which technology we should utilize. Then after that, how to develop a kit, means how to standardize the test for optimum performance of sensitivity, specificity, accuracy, and clinical parameters. And then the validation, and last but most important, to fulfill the regulatory challenges, to make a quality product which can surpass to any international standards. So whole these years, we have tried hard together with our expertise. We have built the R&D facilities, then manufacturing firm at Lonavala, which is FDA approved. And I think when COVID came up, whole this year's experience helped us to come up with a Made in India first kit. And the main focus being an innovation of my lab. So I would request to go for the next slide, please. So I would like to share our long story, but in a brief manner. So when COVID had arrived, the first case detected in January in India. And as you know, the India had to look out for its huge population. The situation was very scary. It was a novel virus. Though the virus was novel, but we have prepared to detect it because we have developed the complete process for development. So that processes, that experience help us to come up with a cost-effective kit, which is quality product with a very short time span, which was the need of an hour. So when India needed a test, where India was importing it from other countries and which has very short supply, the cost was very high, like 4,500 rupees. And just two government institutes, mainly NIV being most crucial for processing the COVID samples. And if you know, if you can see, there were very little infrastructure in India to support the RT-PCR testing at private firms as well. So at my lab, we came up with the first RT-PCR test with all our expertise in very short term, We have designed the test, we have developed it, we have validated it, and got the commercial approval from government as well. And we came up with the test with a cost-effective manner. Initially, we started with the single place RT-PCR test, where screening and confirmatory targets were separated. Slowly, we have moved to the duplex PCR and slowly then multiplex PCR. So in a whole journey, we have come up with improvised products which can give the results in short time with a good quality and which is cost effective. All these things, we were not the only one, though the first one, but I would like to emphasize on the government support and government strategies. Here, Indian government pushed all its might to create and aid the innovators. Government has planned for strategies to manage this pandemic very well. Government supported us to come up with the test kit in a short time, supported us for the funding. Next slide, please. So together, we came up with the solution to fight against the COVID-19. So this was a little story of my lab and of course for India. As you know, the India is very resource constrained nation, but then also with the expertise, with the hard work, with the faith, with the trust and going together, we came up with the solutions and fight against the COVID very successfully. But the main point is, what did it really mean? Means what we have done during COVID, that is one part, what we have learned during the COVID, that is the most important part. And then now with these experiences, what we have to do or what we are planning next, that is the most crucial part. So I would request for the next slide. So I would like to tell you that for my lab, innovation is the whole 9 yards. What does we mean by innovation? Innovation is just the giving something a unique idea, developing new product. No, with this experience, we have learned that innovation is more than just doing the research and discovering something new, but real innovation means to be prepared for hard problems, to understand the pain of the people, to take a solution to the common people, to reach the people, which we can give in a positive way, to impact their lives. So with this approach, when we looked back now, what we felt that we were prepared with this innovation theme in our mind. So we were prepared because we always believed in gold standard, that is RT-PCR test. So even before country was ready to fight against COVID, my lab came up with the test kit. Then the main focus, what we had and we have and we will have it to make the solution for people. So we always wanted to test the kit in very affordable manner because the India is like not very rich country, and here middle class, lower middle class population is high. We have to reach the people and we have to provide a mass screening approaches. So we came up with a solution which was very cost effective compared to international kits, and further we have reduced the cost as we scaled up. The third important is, when the solution is ready, we have to reach to the people. Only solution, only kit is not sufficient at all. That what we have learned through this pandemic, when we have designed and delivered the kit till into the northern and southern part of the India. Again, to overcome the richness to the common people, we came up during the second wave. As you know, the first wave we have surpassed, but during the second wave, the scenario was very bad. Everyone is scrambling. So during that, we came up with the mobile laboratories where we could have reached to the very remote areas and support for testing. So in mobile labs, we came up with the automation system. So automation was the innovation by my lab as well. So we have a lab in box, that is our compact Excel automated system, where we can get the isolation and PCR in a complete manner. So sample to result will be in an hour test. So we have come up with the mobile labs. Then we have came up with the self-test. As the thinking and looking to the pandemic, we have to come up with a very rapid test, point of care test to come up with the solution and for mass screening and to reach each and every individual. So we have come up with the antigen test, which is rapid test, lateral flow test, and with this the software of based on artificial intelligence, so that only single 1 minute test for taking the swab and put it sample on the strip, and in 15 minutes we get the results. It is like fantastic, and then we reached at each and every door. So for this country with a huge diversity, we feel that, and I'm proud to tell you that my lab was front at the to fight against the COVID. So next slide, please. So this is what we have done, but now we don't want to stop. We want to move ahead. And with this, during whole this our journey, who has helped us, that meant us a lot. With a single-handed, we could not achieve this success. We could not support the system. We could not support the society. It could have happened due to support from government, from our advisors, from investors, suppliers. So basically together we did it. So as proud I feel that we are to make the testing available in India, which was the need of an hour. We have to see who has supported us. So the first and foremost was the BIRAC, funded by DBT firm, which is the Indian government, who has come up forward and helped us to scale up our capabilities for production. So it was a huge support we have received from government, which was really helped us to go further and further and to do more and more for our society. Furthermore, everyone came together, like our advisors, our investors, raw material suppliers. Of course, one thing I want to emphasize here, to have a good production scale, the raw material suppliers are equally important. And then we have found it out, the indigenous suppliers only. In turn, we can save the money as well as time. So now we are proud that India has all, like there are different suppliers who can provide us the raw material, and our product is actually an indigenous made in India product. So everyone came together and helped to ensure that India has all the tests which they need. Furthermore, there were so many unknown angels who have helped us during whole this journey, like Postmaster of Pimpri Chinchwad, who had his staff available for us 24/7 to ship our kits on by road to any part of the country. The driver drove more than 17 hours to reach northern or southern Indian territory and handed over the kits. Because without reaching to the people, we could not have supported them, and it was not a complete solution. So we always believe on a complete delivery of the product to the society. Next slide, please. So now, with this whole experience, with together we had done a very good job, and we want to go ahead and ahead to support our people. Now, what we need to think about it and what we need to develop about it. So now for all these things, we have a very good energy, and society has shown, government has shown faith on us, so that we can do more and more developments. We can learn more and more technologies to come up with the solutions and to make the India independent and world-class brand, which is made in India. So now our major focus will be on point of care and automation. So diagnostic kits is one part, but to test it, it will be accessible to the society. It will be feasible. It is very easy and have a need very little infrastructure. Then only we can reach to the actual people. So for that now, we are trying to develop ecosystem, where recently we have acquainted the company known as Swayam. Basically, this is a health chaos, where we can tell it as, like we can say it as a healthcare ATM. So in 15 minutes, we can test more than 15 tests of blood test, liver test, kidney test, so that, and it is a like point of care, and we need not to have any expertise in it. So with this experience, with innovations, with various approaches of going beyond and beyond to our territory, we are focusing on point of care system, which is a right path to the diagnosis, innovation, and production scale up, and to reach the people with solutions. So for whole this journey and our future perspective, though we are trying to reach at international way to each and every part of the world, but our main focus would be to serve people, to help poorer, because we have also came up from India, which is a resource constrained country, which is a little bit poor country. So focusing on the Vasudeva Kutumbakam, we want to help people with economic solutions, with innovative approaches, and with accessible solutions. So
you can wrap up, Minal, please.
So next, please. So with this story, I would like to thank you, where together we can do, we had done, and we will do. So being a MyLab person, I would like to emphasize one more thing, that the MyLab is not the organization, it is a family. And being Indians, I'm really proud to be part of the MyLab, to be part of the India and made in India. So we want to support our country, we want to support our nation, and not the nation, but we want to make a brand, made in India brand, for whole the international countries. Thank you so much.
Well, that's lovely. Thank you again for a very inspiring talk. I think many of us listening would applaud you from wherever we are around the world. So I now open it up for questions. Barbara, we had some both from the first talk and some from the second. So do you want to select a few to raise?
Yes, so first, Dr. Minal, thank you so much for your presentation. And there is a question on the chat. Is there anything published on the automation and AI you mentioned? Where can we get more information about this?
Yes, we have our own site where we have published about automation and AI, so that you can get a little bit updates from our MyLab site. Secondly, regarding the automation, we have one publication as well for NatSpert, where we have developed a complete automated system on Compact Excel platform. And if, in that case also, if you would like to know more about it, I will share all the details with you.
Thank you so much.
In brief, I would like to tell you about our automation system. It is basically a Compact Excel automated system, completely developed indigenously by MyLab. Since 2016, we have started working on it. Here, it is a 32 sample testing system where we have to load the clinical samples. For example, say respiratory sample in VTM for COVID-19. And then the machine will automatically do the extraction part and master mix preparation for real-time PCR testing. And second is real-time PCR, where amplification and detection will be carried out. So right now there are more than 70 installations all over the India for Compact Excel, and the COVID-19 testing has been done on Compact Excel as well. Even in mobile vans also, we have launched the Compact Excel. The mobile van is a project which is in collaboration with ICMR, and it has been deployed at Delhi Airport, Mumbai Airport to have a testing for international travelers, and even at remote areas like UP and Bihar, so that we can reach to remote and help people.
Thank you so much. And there is also another question for the first presentation, Dr. Mukherjee. How was the large consortium formed and coordinated, considering the varied interests of many parties?
Thank you for the question. I think the first and foremost thing here was, we were passing through a national emergency. That, I think that was the basic idea, and everybody got together the manufacturers, the raw material providers, the importers, and the government. So we created this platform, and so that all of them can come into this platform, get their raw materials identified and sourced, or indigenization of it. So it was first and foremost, nobody, and that is the beauty of the system, was every innovator, every industry which came together kept their personal the interest of our organization at side, and looked at the interest, how good we can bring out a kit, how fast we can bring it out, how affordable it can be, and how it can be maximally used. So that was the basic ethos of creating this platform, and that helped us. Of course, you are right, there are a lot of corresponding interest comes up when you're really working on a business platform. But here that was a bit, I think, thanks to the innovators and the companies who really worked on it. So that's how it was. A board was created which had representatives from the industries and everybody who really worked it out and helped them to channelize raw materials and at a very affordable and a structured cost. So I think that was the ethos which was built up, and really got the support from the startups and the industries and the companies who really helped up to move it. So that was the idea of how it was brought up with varied interest, and it was really crystallized and taken up.
Thank you so much. I can see that Michael raised the hand, so maybe there's a question there.
Yeah, I think I would just like to commend you on the framework that you created, because something that we encountered with dealing with many other countries is that there's kind of, people don't want to develop such a framework because it means that often there are lots of vested interests in these systems around diagnostics because there's so much money. And it's like, as soon as you have a framework where things can kind of be equitable and everyone has equal opportunity to develop diagnostics, then people get a little bit edgy. And so it depends on who is like controlling the regulatory framework in the country. And so it's really nice to see a good example of someone who's essentially doesn't care about like all the vested interests or whatever, or maybe you just work within a system where you're actually able to make these decisions without so much pushback from everyone else. But yeah, it's a good example for other countries, I think.
Sure, absolutely. And we are also very happy to catalyze it. And the government is really catalyzing it. And and it is not a one-way traffic that we decide and catalyze. It is a equal mutual cooperation which we get from the innovators, the companies like MyLab and other companies who are literally supportive of such type of initiative and come together. That is how it goes on. So that's the idea. Over.
Thank you. There is actually, Keith, there was one question from you. At which point in the development cycle does Indian industry investors typically engage with the medical device developers? And how would you characterize the risk?
Minal, would you like to answer, or I should answer first?
So you start being the most senior person and most experienced.
No. So I think, as far as investment from our side is concerned, we looked at, how good is the team, how affordable, how strong is the science, how good is the team, and how quickly we can develop up, providing all the innovation into place. So that is our concept of investment into it. And we invested proof of concept, we invested at a later stage as well, and till product commercialization as well. So we invested with that in mind. Now the angel investors or startups, I think we should really thank them as well, because they also invested looking at public good during this COVID time. Of course, their return on investment has to be built in, and they had built in that. But public good was one of the foremost things in mind, because we were looking for kits which really wanted to be literally quickly developed, validated, and deployed. So that was the thing. And I think Minal can answer, because they must have handled much more investment than we have invested. So I think she can. But I think the ethos was once again there, that public good was in mind. Over to you, Minal.
Yes, sir, you are absolutely right. First of all, I would like to thank you, sir, because at DBT, BIRAC was the first government organization who has supported us to scale up our production during the initial lockdown period and when we have came up with the test kit. See, only developing is not the solution, but to deliver it with the right time, with the right mass is more important. We feel that. And without without investors support, we could not have done whole this journey with a successful outcome. So investors and investment is really important. And from our perspective, like from the manufacturing firm perspective, we have to prove that our product is a quality product and it is meant for the public health.
Lovely. Thank you. I have many follow-on questions, but I hand the floor back over to Barbara. Thank you.
Yes, there's one more. Yes. So, Dr. Mukherjee, thank you for the presentation. Which sector led to the platform framework? Is it the government? And for significant decisions redirection, which sector has most push?
Can you just slowly elaborate the question? I missed some part of it. Can you slowly elaborate it?
Okay.
Yes, of course. So which sector led that platform framework, and is it the government? And for significant decisions redirections, which sector has most push? Sorry.
Okay, I got it. Is in the chat as well. So I think, yes, the government catalyzed and initiated that platform. The framework was created by the government. Of course, government, once it created, it has some convening power. So always, I should not say a dominating power, but a convening power per se. So people will listen to me. If I call Minal will definitely pick up my phone, because being in a government, she will definitely, structure we have it in India, and a respect for the government people have in India. So definitely we have a convening power. We created the platform and the framework, but we brought in equitable stake from the end users, the companies, everything had, and the decision was not ours. When the decision was to be taken, the decision was taken by the group which was created there, or which a structure or a decision-making body which was created there, which had equal representation and majority representation from the startup innovators companies, where the major representation, they took the decision. We never interfered with that, and that is how it is. That is how the push was created, and it was catalyzed very fast. So we just created that platform, and through our convening power, so we were there. But I think the major decision, everything is taken by the platform itself. Over.
Thank you so much.
I had a question. You mentioned at the beginning telling a story, and you mentioned a fairy tale. And I wondered whether, and Minal described diagnostics at a quarter of the cost and accessible and accessing the poor, which I think resonated with so many of us. How close do you think we are to reassured or the assured criteria for a quality diagnostic? So Minal, you mentioned focusing on quality. Is quality and the WHO assured or reassured criteria of affordable, accessible, simple, sensitive? Is that a realistic objective in the next few years, or do you think it remains one of your fairy tales?
So I think it's a very, very interesting and very important question. The fairy tale, what I meant, and I really love that analogy when I gave it to my prospective grantees, when I do my grant writing workshops or grant writing clinics, is that you need to bring out a product. It may be diagnostic or a medical technology device, when anything which has to be affordable, accessible, best in quality, And best of the quality means it needs to be highly, in case of diagnostic, it has to be sensitive and specific and has the right specificity as per the TPP, the target product profile developed by the regulatory agencies. That is the end game. You can't bypass this, and I think I'm very happy, very happy to report that all the diagnostic kits, including the one which has come up, they literally, including the MyLabs one, has literally been into the quality standards, has built up the quality standards and have passed all the quality tests onto it. Whether internationally, of course, every company will like to export it. I will like to generate more business and get WHO pre-qualification wherever and WHO standards wherever it is needed. So they will aspire that, and that is essential to expand their business. And but Indian regulators are very, very sharp and strong. You can't bypass any system, and so ideal will be to get all the standard in place. And I hope it doesn't remain a fairy tale. It literally gets into the right perspective, and the companies are aspiring for that. Over. Minal, you want to add something?
Yes, sir, as you correctly mentioned, for quality, we have to go with the international standards, and for regulatory, without regulatory approvals, not a single diagnostic kit can come into the market.
All right. Are there any other questions? We're running a little behind time, but one of the speakers for the lightning talk has unfortunately had to drop out, so we're not running too far behind. Any last questions? If not, I think we'll take a 5-minute break. Come back at 5:15 Singapore Philippine time, so that's about 7 minutes. So a lightning break before the lightning talk. And just leaves me to thank the two speakers. I think you've seen the comments. Everyone is very impressed, and we watch with great interest. And thank you for sharing.
Thank you very much, Jennifer, Barbara, and Keith, and all of you for listening to us. Thank you very much, and namaskar from India.
Thank you so much.
Thank you, team, for this opportunity, and it's a really very great discussion among all of us. So thank you so much.
See you back in a few minutes.
Oh, Keith, you're muted.
Try that one again. Welcome back. Can you hear me now?
Yes.
Okay, all right, there you go. So now we move into a series of lightning talks from around the world. I'm not sure if we have 3 or 4. The first is close to home here in the Philippines. And then we have one from the UK, one from Indonesia, and then possibly one from Cameroon. So without further ado, I'll first introduce Kahlil, a colleague down a little further south here, who's going to talk about setting up local production. Without further ado, over to you, Kahlil.
Hello, everyone. So I'm just going to quickly share our experience during the COVID-19 pandemic and why we are aiming for local production of some basic lab reagents. So our experience during the pandemic was quite, it was outside the usual institutions of where science is usually done, because we were an international volunteer team, and we all saw a common problem, which was that while we know that we need to sequence SARS-CoV-2 to keep track of its more virulent variants, we also knew that we didn't have enough sequencers here in the Philippines and in the rest of the developing world. So, but we were sort of not a poor nerds, so we thought, hey, why don't we just send sequencers to labs, maybe start with the one lab and then organize trainings for them. So that's exactly what we did. We raised funding in this open science platform called Just One Giant Lab, and then we got help from scientists from all over the world. And so March of 2021 was the first ever sequencing of SARS-CoV-2 here in Mindanao, and I believe it was also the first SARS-CoV-2 sequencing all over the world enabled by a volunteer team. Normally it's like institutions or governments. So, but from that experience, we uncovered a more fundamental problem in doing molecular biology in the developing world. And all of you are familiar with this, which is that, so if we buy reagents from the US and the UK and Europe, it's like, it's already expensive for us, And then second, if you need to transport that halfway around the world, there's additional costs. And also getting through the bureaucracy is also expensive for us. So it just adds up to the cost of doing science in the developing world. So, and I'm preaching to the choir here, we could skip all of that if we do local manufacturing. And now is the time, because you have open source wetware, you have some lower cost hardware, and then there's the pandemic just gave us this, made a lot of people see that this should be done. So that is what we are hoping to do. We are working with Beneficial Bio. We are working with this Argent team called Friendzymes to make this happen. We just got the donation of a Opentrons fluid handler, and it's still a lot of work, but the components are there. I talked with Keith about this. It's just makes sense to do it. Yeah, so that's basically what we're hoping to do. We're hoping to work with all of you to make this happen, and also maybe help other areas as well with the same problem.
Lovely. Any questions for Kahlil? In particular, I suppose I had a question around the organization. Maybe it goes back to the discussion earlier on about international groups. There's a map I've seen of all of the component players in, is it Friendzymes or Beneficial Bio? I forget which. And there's lots of people from around the world. Coordination must be a little awkward, a bit difficult, but presumably that's a strength as well.
Yes, it was it started as accessible genomics. Yes, accessible genomics, and it just came from that. So now that so that is continuing. but we are expanding to this more fundamental problem of access to reagents. So what we're looking at now is to, well, in terms of legal, as a legal entity, so we have existing collaborations from different people in different labs as well. So we're looking at the future, looking for grants. That's first. So to fund this, and then also to set up legal entities which could further look for grants and apply for grants and be the nexus of like collaborations between different labs. So the other one is to make this sustainable by setting up a corporation. In the Philippines, I think there's no like a social enterprise corporation, it's like a regular corporation, to make it a sustainable business. Meaning, we want to replicate what Beneficial Bio has been doing in Cameroon and Ghana, and to move forward and to sustain this operation, this manufacturing operation and its development through selling to local labs and also maybe local distributors. So that's what we're looking at. It's very early days, but we're one thing what we've been doing during Accessible Genomics and this endeavor as well is working with the garage door open. So we're documenting in the open what we're doing. Just go to biokhalil.substack.com and we're just documenting what we're doing there. This is what's our problems and how we're solving them. It's all there. I'll put it in the chat.
Yeah, please do, and then we can add it into the chat notes. Thank you. Moving on, thank you for that, Kahlil. Michael, back in the UK, so my two homes, Philippines and the UK in one go. Over to you.
So the thing that I'm going to be talking about just quickly is the development of kind of open standards and RNA extraction protocols. So something we found out at the beginning of the pandemic was that there was poor availability of clinical samples to do your validation. And especially we were an academic group, so we didn't directly interact with the NHS to try and develop diagnostics. So we developed our own kind of MS2 phage that we packaged with coronavirus RNA. And so we were able to use that as a kind of a process control for our entire process from RNA extraction to PCR, and then go directly to patient sample validation. And we showed that it was a good predictor. It worked really well as kind of predicting whether you would get the result that you expected with patient samples. So we've made these materials available under the open MTA on AdGene, and also available for me if you would like to reach out. We're kind of growing the library of materials. So we've also I've just put on AdGene, with the different variants, with the different WHO variants, I now have plasmids that would allow you to make these virus-like particles for those different variants. It's a smaller portion of the genome, it's not the whole genome, but it's enough to be able to distinguish between the different variants. And I'm just about to write up how you would make these particles and package them with DNA instead of RNA, because they're natively packaged with RNA. And then this would be a kind of a simple low-cost protocol to package them with DNA. And all the materials are available under the open MTA, so free to commercialize with what we've published.
That's fantastic. Any questions for Michael?
What we've also done is we've taken part in the coronavirus standards working group run out of Stanford. So we've been able to get an international unit equivalent of our standard compared to the WHO standard. So it's been included alongside 7 other commercial standards and tested in 14 different laboratories to kind of get an idea of how reproducible it is. Because there was a lot of criticism around exactly how, well, the world has not done very well around COVID testing. There are no standards. Everyone shows off like CT values and things like that don't mean anything in isolation. And it's not clear how you can make any clinical decisions on kind of qualitative rather than quantitative results.
And experimentally, how simple is it if someone was to get the constructs matching or from yourself? At what sort of scale can people make these? I mean, can you use them? Can you transfer it into a manufacturing standard, or is it remaining a sort of a research use only?
I mean, I think
laboratory standard.
So in general, if you do, it's just like a his tag affinity purification, and you would get more than enough for hundreds of plates. So in general, what we do is we aliquot into 96 well plates, and then you can just use one aliquot at a time. So it's like a pop out plate. You just pop out a single aliquot, and you would get hundreds or even perhaps thousands of plates from a single purification. And that's 200 milliliters of bacterial culture. So you could easily scale it up further. And I don't think it's too tricky to then do the manufacturing if it was. The idea is to make this kind of distributed manufacturing of standards. So you could have a localized sensor, which basically does the manufacture, quantifies it, aliquots it, and then you can distribute it locally to kind of support diagnostics in those areas.
Lovely. People may be giving you a call. Any other questions for Michael? That was very interesting. If not, we jump back to, yes, can you paste the agenda,
yeah, I'll do that,
please? So if not, we jump back to our final likely talk to Susanti from Indonesia, who's going to switch gears, I think, and talk to us about colorectal cancer, something close to my heart, for sure. That's the reason why I'm here. So, Susanti, over to you.
Hello.
Hello.
Hi, sorry. It's a bit glitch there. Can you hear me well?
You can.
Yep. Okay.
Yes, you can.
Thank you. Yeah, I'm Santi. I'm CEO and founder of Diagnostic Technology. So basically, we are a startup based in Indonesia. Can I share the screen a little bit, or is no time for that?
We have a few minutes, I think. Please, if you can help.
Yeah, I think I have a video to run, and then just to give a little bit of background, and then yeah, so here we are. So our value proposition is molecular diagnostic for all. I know it's kind of like big dream, but we want to achieve global equity in precision medicine by providing low-cost molecular diagnostic platform for cancer. Run a little bit of video.
My name is Santi. I am a cancer researcher. In the middle of working on my research, I was diagnosed with stage 3 colorectal cancer. At that time, I had so many questions. Why me? I'm quite young. Then I remember my cousin was diagnosed with cancer too. Is this disease inherited in the family? Are the drugs given to me effective? Can I be cured? How long can I survive? I believe many others with the same condition as the one I have ask the same things. As a cancer researcher, I knew that the treatment for cancer patients varies from one patient to another depending on the genetic mutation profile of each patient. Thus, molecular diagnostic tools are very much needed to be able to guide doctors to provide appropriate and useful therapy. It is also important to understand the familial risk of the disease. Unfortunately, molecular diagnostics is still difficult to reach by the majority of cancer patients in many parts of the world because of the high price in the market, including in Indonesia. Through excellent research and innovation, Pathogen Diagnostic Technology would like to provide affordable molecular diagnostic solutions to support better management of cancer and other diseases in developing countries. Driven by our young and highly enthusiastic teams, supported by world-leading experts in the field, Pathogen's vision is to democratize molecular diagnostics to achieve global equity in precision medicine. Pathogen, molecular diagnostics for all.
Yep, that's a little bit about us. Sorry. So basically, we did a little bit of market survey, and we know that at least in Indonesia, only 18% of healthcare institutions have an access to molecular tests for cancer, for guiding therapy and also detecting familiar risk. And while we know that cancer treatment is expensive, molecular diagnostic is very limited. Meanwhile, it's very important to make the cancer treatment as efficient and with higher efficacy as possible, because the money is tight and the cancer treatment is expensive. We don't see that next generation sequencing is feasible in many developing countries in clinical implementation. And we captured that there is a global increase in PCR machine and related equipment during COVID-19 pandemic, and therefore our solution kind of become pretty relevant in this context, because we're providing PCR-based molecular diagnostic kit for colorectal cancer, ColonMelDX, and for lung cancer, PulmoAmDX. So it's PCR-based it can be used to predict therapy response, familiar risk, and then prognosis. To accompanying the PCR-based test, we also developed the digital pathology platform to examine the protein biomarker. This kind of the impact that we love to achieve. So we would able to prevent under and over treatment of thousands of patients per year for the, colon cancer and lung cancer in Indonesia. Only this number, but then obviously it's higher if you look at the developing country, other developing country, reduce ineffective therapy, as well as early cancer detection for the family with the high risk. Yeah, that's it. We are located in Indonesian government research agency, Center for Innovation, Science and Technology Department, Indonesia Institute of Science. Thank you very much.
Well, thank you for that. It's a timely reminder that the world is dominated by COVID, but there are so many diseases over and above COVID. Are there any questions or comments that anyone has for Susanti? I had one specifically related to mutational analysis. One of the challenges of doing cancer diagnostics, of course, is the whole range of mutations that you can get. And the question is, of the very large number of mutations, which ones do you target? Do you target only the most common ones? Do you try and be broad or focused? How have you tackled that question of what actually to try to detect when it comes to, let's say, colon cancer?
Yeah, thank you very much, Kate. Indeed, cancer is basically genetic disease, and every day I think we found some potential genetic biomarker. So our strategy right now is to focus on small panel that already clearly have a clinical significance. So in colon cancer, for example, we need to start somewhere, and I think a small panel is the most reasonable way to do. First, to increase capacity, so to test the water, increase capacity of the local clinicians, laboratorians, and even the patient, like raise awareness how important these genetic markers are. So for colon cancer, for example, in our kit, we have MSI, microsatellite instability. This is very important marker in colon cancer. And in the UK, it was compulsory, I think, the clinical guidelines suggest that every single colon cancer patient need to be tested for MSI. And this, for example, MSI can predict response to 5FU, the most common regimen in Indonesia. And we know that at least based on the data from the West, 15% of colon cancer will have MSI, which means it won't be a response to 5FU. And then MSI also can inform about immunotherapy as well as a targeted therapy like cetuximab. And we have another important oncogenes like KRAS, BRAF, PIK3CA into our panel. But yeah, that's how we go. Small panel, very, very strong clinical significance.
It reminds me a little of Minal's approach, which is starting in one area and then progressively building, I think, when she told her story. So thank you for that. Barbara, we had one question from Jenny.
Yes, so thank you so much for your presentation. And there's one question: what are the challenges you have found in setting up your startup in Indonesia? And is there any advice you would give others doing the same?
Yeah. Startup, so I'm I'm actually cancer scientist. So startup world is obviously something that pretty new to me. So the challenge from the business side, like from about the establishing the startup is, even though I'm Indonesian, but I mean, like, I'm located in the UK right now, but I'm Indonesian. Even for me, the regulation pathways is confusing still from business side as well as clinical side, because obviously our business, biotech, molecular diagnostic, is heavily regulated, but then in developing country, I guess, it's not exclusively to Indonesia. Sometimes it's not that clear, like where you can get the information for certain regulations and things like that. That's one thing. And then second thing about the funding for the startup. One of the reasons I think why biotech resilience is not strong enough in developing country is because lack of funding, and it's translate also into the business side, like for startup funding. It's very few investor interested in doing this, investing in this biotech company, because they usually invest more into fintech or into, if they invest in health, it will be like telemedicine or something like that, you know. While biotech company, we need a lot of money, quite relatively slow in terms of turnover, and then not that strong support of regulation as well. So it's very risky for investor. So the funding also a challenge. And I think the third is the consumer. You need to make your market ready, basically. Like us, it's very limited molecular diagnostic for cancer in Indonesia in terms of clinical implementation. So right now, we constantly, before as well, like we constantly do a lot of workshop, capacity building, so they're ready. They know how important it is. They know how to do it, and patients also have awareness about this. I think those are some of the challenges. So if there is any advice give for others who want to doing it, I think similar to many other things, if you have a passion about it, you will put 100%. But obviously, you need help. I got the support, lots of support from many people, not only from scientists. I got support from the business side, blah blah, and government as well. Engage in developing country, engagement with government and policymaker, I think one of the key. So yeah, I think that's what I can say for now. Thank you.
Well, thank you for that. I think several of us around the call will resonate with those three responses you just gave. But I think that's part of what Reclone's about, trying to help address those three things. So thank you for each of the speakers for their lightning talks. I appreciate that. So now we're going to maybe have a more informal chat with those who want to share their thoughts and continue discussions. There are 3 breakout groups now coming up. Kahlil is going to discuss the supply chain. Jenny, I believe, will be taking over the discussion around manufacturers, something which is part of the discussion. And it's nice to see a couple of students still with us. And Aish is going to talk about networking for students in diagnostics. The earlier we start to train researchers, I think, the better. So, okay. So Jenny, do you want to sum it up then? Do you want to wrap up? I mean, my take, my 2 cents were, or 2 pesos, were just how integrated the first book was. All of the components were brought together. It really is a holistic approach to the diagnostic problem, which I thought was fantastic across academe, industry. That was the thing that struck there. And in the second talk, it was just the sheer determination to succeed. They had a clear vision as to where they wanted to go. It was commendable, delivered a clear strategy and delivered on that strategy. Can be done, obviously with the help of the government and the advisors, which seemed essential. So I think it gave us all hope that what we're all aspiring to is challenging for sure, but not impossible. So I'm so glad that we had those first 2 talks.
Thanks, Keith. Yeah, I think they're really fantastic talks, and we will put them on YouTube. And if you think there's anyone who'd be interested to look at them, do share the videos. As you know, many of the Reclone community are not on a friendly time zone for this meeting. So this was our first one to make Keith not stay up really late, and Kahlil and others who usually join us extremely further into the evening. So thanks so much for making the effort to come along. If you found this useful and you think it would be helpful maybe in 6 months or so to have another kind of one at this time zone, do let us know. We feel like we didn't quite reach out as far as we might have done. We sort of made a concerted effort to contact certain researchers individually kind of in the run up to the event, and I think that was quite successful, but probably a bit last minute on our part. So we will try and do some more proactive reach outs to people who are working on this type of issue in Asia Pacific who may not, specifically of course with Reclone's focus in LMICs in Asia Pacific as well, who may not otherwise have come across Reclone. But if there's anyone you would like to direct to the network, please do that. We, as you know, we kind of have these symposia on an ad hoc basis. So the next one will be probably in around 2 to 3 months. In the meantime, we have our fortnightly Reclone community calls, which unfortunately are less well timed. But again, we can maybe weave in a sort of Asia Pacific friendly time zone as well, because it's every other week. So that's fine too. If you have any suggestions of speakers who you'd like to perhaps do a community meeting, get in touch with Barbara. She can invite them. And we have one this afternoon, actually, for anyone who does want to stay up late. So I think that remains that we can leave the rooms open for networking. Obviously, there's not that many of us, so we'll just leave the main room open if you wanted to have a chat for a few minutes. Otherwise, this is the end of the formal proceedings, and you can head on and go about your day. I'm just
really want to go through those first 2 talks and digest them and think of it a bit more. I just feel like there's a huge amount of experience both in manufacturing. I remember back when I was in Glaxo and Merck, we were always mindful of how good India was at manufacturing generics. Of course, when I was on that side of the table, I didn't like it very much, but of course now then tables have turned. And I just wonder whether they are reaping the benefit of some of that just engineering, production, chemistry experience. And turned it to vaccines and turned it to diagnostics in a way that we just don't have here yet.
Yeah, I think definitely the background. I sent a private message to Michael during the talk as well, that I think it's interesting to contrast that to, I mean, India obviously is a bigger country, so it has a bigger biotech industry than South Africa, but in essence, both of them had indigenous capabilities to manufacture, and yet very different stories. And so I think that I'm just really impressed by the way that Shirshendu and all of the others involved in that initiative kind of actually looked at what the roadblocks were and tried to remove them. And
got so many people to buy in to a common aligned vision. It was there was that circle of diagnostic. He had one slide. He only flashed it up briefly, and it had a circle of all the different. And I looked at it and thought, yeah, those are the 15 different pieces of the jigsaw that you need if you wanted an integrated diagnostic solution.
Yeah. And they were pulling in the same direction, not in opposite directions, or kind of, yeah. And I think that made a big difference. It was,
yeah, one of these days I should jump on a plane and try and go and just see what I can learn. I know JJ who wanted to stay longer so he's my handler, my funder, everything. I mean, he was, I think, taken certainly by that thought. That's exactly what he's trying to build.
Yeah, it's a really, I mean, I kind of have seen Shirshendu talk briefly about it. I hadn't seen the full exposition before, so it was really good to see that. But interesting, I mean, I don't know if you've seen the news today about Cuba and their vaccine. I mean, Cuba's another place where, because of all the trade sanctions with the US, they had to develop an indigenous industry in biotech, and they were very strong, and like they invested a lot in biotech in the sort of 70s through 80s. And sort of obviously traditionally have been very well renowned for their health system. But there was a bit of a, well, from the perspective of researchers, I've seen some opinion pieces that kind of post 90s, like in the 2000s, the investment levels started dipping in biotech, but they still have retained that capability, and they've just released really good clinical trial results for their own vaccine that they've been developing in Cuba. I don't know the situation with Diagnostics, but I'm guessing that they did also manufacture a reasonable amount of diagnostics. I think it's an interesting kind of case study in what happens when, yeah, a few decades ago, you sort of push on capabilities and then reap the rewards at a certain point. I mean, well, who knows? There's a long route to go, but yeah, Cuba's an interesting example.
If Michael's still there, isn't Kappa Biotech a South African company, a South African biotech company, who make enzymes for?
Yes, they were bought by Roche.
Roche, gosh.
And the problem, no, so I mean, it was Kappa Biosystems, but they were bought by Roche, and loads of people made loads of money out of that, except the government that actually funded them. So it was like I think it was a spin out of the CSIR that started making enzymes. And it was quite controversial because the TIA, I think, invested quite a bit of money in them, built up all the capacity, and then made no money out of the sale to Roche. And now there's another company which is, what's it called? There's another company making enzymes, but it's kind of like they start from scratch again. It doesn't make sense. Like Kappa was, like people still use Kappa for next generation sequencing library prep. Like it's one of the best enzymes that you can get, but it's like now South Africa's decided you have to start from scratch again because obviously they've sold off this company. And so now there's another company which is now doing the same thing, or now doing like diagnostics enzymes. But yeah, it doesn't make sense.
Yeah, Cape Bio. Daniel Nadine Reclone, and he's also talking at this MSF event. He's the first speaker on Friday as well.
Yeah.
But yeah, I mean, basically, it's, again, like Michael said, started, as far as I can tell, from scratch as a startup with, you know.
Which doesn't make sense because all the expertise was there from the Kappa. Yeah. So like, what is the point? I don't understand the.
Yeah, but did Roche keep the manufacturing site in South Africa, or did they move it to the US? I was struggling to figure out if this manufacturing site in South Africa was still active.
I don't know.
Well, so I asked that because for the first 6 months of our project, we were using Kappa, mostly because it was the cheapest one, because we didn't have any money. And everything was going all right, except for the fact the supply chain stopped. And they Merck just told us one day, so first of all, we have 3-4 months delay time. And so you wait 3 or 4 months and you don't think of anything of it because, well, it's just normal. But after 5 months and 6 months, you start asking. And they said, oh, it's not going to come. The supply chain has stopped. And so I wondered if there's something that happened in South Africa to cause problems, or whether it was just Philippines wasn't important enough, you know, and all the supply was being used by other people. But
this is similar to what happened with RPA when Abbott bought whatever company it was.
Twist DX.
And then they kind of dismantled the company. So then outsourced the manufacturing to another company in San Diego or something. And so I think like RPA will never catch on because there's no way to really scale the manufacturing meaningfully, I think. And it was also like you always had variability in the manufacturing. And I'm not sure, Abbott might have just made the acquisition to literally dismantle the company.
Fire and extinguish the Microsoft approach. Yeah, the founder of TwistDX has now gone on to start another company, still doing RPA stuff. So he must have retained or is licensing his original IP in some way. I've forgotten his name, but yeah. But I came across him because his RPA was written into an like a funding application for local production at a site in Africa, and which he'd clearly been involved in kind of like feeling. But it just seemed like if you're gonna, I mean, this was for a site that was starting to do molecular manufacturing. You're starting with RPA. I mean, this is one of the most complex mixtures.
I think a lot of people made a mistake with COVID around picking LAMP and RPA in a sense, because it's like the regulatory system was not ready for those kind of different approaches. And it's like, just get qPCR right. qPCR is bulletproof. Like people say the equipment is slightly expensive, but it's like once you have a qPCR machine, you're pretty much set and ready to go.
Yeah,
and you kind of need the same thing. Even for LAMP, you would need something that could detect fluorescence anyway, or like absorb it. So I think people really missed the boat with that slightly, and it made far more sense just to focus on, like, to really have an impact with this. It made far more sense to focus on manufacturing qPCR enzymes.
So, well, I think I slightly disagree. In a country where 3 million pesos is a lot, or $50,000, whatever a PCR machine costs, even the cheapest one, it's not something that is easy to get hold of. The other problem we had is that what's the point in building a kit that already exists and where people can just import from China? And so it wasn't. I mean, we've built both because I'm not sure that the Philippines is necessarily ready for LAMP. Regulatory, it doesn't know what to do with LAMP as a regulatory question. It's not a rapid test. It's not PCR. It doesn't exist. It's just not a thing.
It's also not as sensitive as PCR. So you would say that also.
No, it's not. No, it's not. It's horses for courses. It's a fraction of the cost. You have to remember that, at least for our setting, there are 3,000 rural health units that have nothing.
Yeah.
The equivalent to GP surgery.
I mean, I'm not sure I agree on the cost issue, because I think that at least if you buy LAMP mix from NEB, for example, it ends up being just as expensive, if not more expensive than qPCR.
Sorry. Yeah, I meant on the instrumentation, on the hardware. You're right, on the BioWare is the same. I'd agree with that. That doesn't make a lot of difference at all.
I think it's a timing issue as well. Like, I mean, there's, like Michael said, in terms of impact for COVID, because the qPCR regulation was already in place and the pipelines, it's sort of a lot of the LAMP innovations that people started, like, they're going to take time.
Exactly.
We know, but I think a lot of people who started out on that route kind of had a sense that if we just build it, then it will kind of be useful right now, and the timescales are very long. But one thing, I mean, qPCR, we've talked about this before, Keith, is like access to large scale amounts of dye, because a lot of those are off patent now. And it feels like the chemistry is not beyond the realms of reason for someone who knows chemistry. It's just not my field, but I feel like that's one of the downsides of the qPCR kits. I don't know, Michael, if you've had experience having to buy large amounts of qPCR dye for master mixes, but that feels like the enzymes, as biologists, we know how to do that, but the chemistry side of things, but the reactions are pretty well described in the literature. I think even some of the stuff that's pretty, like Pico Green, which is one of the most sensitive, my understanding is, for double-stranded DNA, that's now off patent, but it's still very expensive.
Certainly the intercalating dyes, the cybers and the cytos, those are all off patent now. Amersham had brought all the patents up when I was a young buck scientist, and those have all long gone. And you're right, the chemistry is not that hard. It's just got to try and find a chemist who
any chemist, just any chemist,
or find a chemist, and end of story. But I mean, it is ridiculous when you work out you spend $1,000 on a bottle of cybergreen or cythinine, and you work out how many milligrams are in there. It's nothing. The markup is well, the markup. I think making the TechMan dyes probes. I mean, that's a different beast. I mean, that's phosphoramidite chemistry. Is not trivial chemistry. But the intercalating dyes to make a simple cybergreen assay is it shouldn't be that expensive.
Do we know any chemists? I mean, I'm in a chemical engineering department. I should be able to find myself a chemist, but
I mean, something that will be interesting is that so DNA scripts, the enzymatic DNA synthesis, they've started to say that they can make probes, the hydrolysis probes.
Okay.
But yeah, I'm not.
And there's a German company, isn't there? German startup who's got one of these nano synthesizers, really small scale.
Oh yeah. I know it's Kilobaser. Yeah.
Kilobaser, that's it.
They still don't make sense unless you're optimizing for speed of like, I need these things now. It's like it works out something like $50 per primer.
Yeah, it's ridiculous. Even script is tricky. It's probably about $10.
Because I got interested because just anything that removes the supply chain for us is great. So
yeah, well, we did too, and we for Cameroon as well. And we looked at the pricing, and we looked at the price of shipping stuff. It's cheaper to order it and ship it.
So that's what we came to as well.
Because I mean, we get stuff from GeneWiz or IDT, get it delivered to the UK. Primers are not that difficult to ship. It's like we just shipped 125 primers. It cost us £70 to go from the UK to Cameroon, and then obviously customs as well. But even adding on customs, they're £3 a primer, so you just can't. Can't. It's not possible to beat the pricing.
Well, anyway, I had better go and see what's been happening. Thank you for an enjoyable afternoon.
Thank you so much for facilitating. It was very nice to have
thank
Barbara
you so much.
Given the opportunity. I've been tweeting, so I was doing something useful. But
all right, lovely. Well, in that case, Michael, thank you. I'll be in touch, and Barbara, Jenny. Oh, Jenny, so we are starting this IPRDC tomfoolery seriously in January.
Oh, great.
So we probably will come back to you and ask whether we have all of the right clones up to date, and whether you have any advice since we did that pilot a year ago.
DNA for you by January. So we're testing the new backbone for the research and diagnostics collection. Sinan Federici in Chile is sort of testing it with Cesar Ramirez, because we realized that the backbone that we used was rubbish. Sorry. We knew it worked as a cloning backbone, sort of, but and it was the one that Free Genes were using. So we kind of thought it's been, there were several collections out in it at that point, but no, it turns out it's not brilliant for assembly, and it just for some reason is just, even though the expression cassettes are okay, like they just don't seem to work very well. But we've tried them in a bunch of other backbones and found some much better ones that are also under OpenMTA and et cetera, et cetera. So they're just optimizing them, and then we'll basically reissue the collection in the new backbone, which should give you, so the cassettes work okay, you just have to PCR them out of our backbone and stick them in a better one. But yeah, hopefully that will be, and then we'll refresh the collection at Free Genes and add Gene, so that the new one is included. Because at the minute, basically, that we don't have stuff that people can just receive and start expressing. They definitely have to take it out and put it into something, and we want to end up with plasmids that are really ready to go. But as a side effect of doing the designs during the pandemic, when we didn't have access to the labs, and also being limited on time and people and like workflows being prepared, it's just it was a unfortunate byproduct of that, but I think we're in a better position now. So we'll definitely make sure you have the updated ones.
Well, on the upside, it was your help that got us the proof of concept, that got us the funding. So all hats off to you. So
you didn't do so much, but you're welcome.
So we hopefully will start that in earnest in January.
Fantastic.
And to Michael's point, we're starting with PCR for a good reason.
Yeah.
So anyway, thank you very much, and have a nice Christmas, I guess, if I don't speak to you.
Yeah. Well, hopefully we'll be in touch by email at some point with discussion of plasmids and things. But yeah,
I'm actually coming back to Birmingham for a couple of months, so
really?
maybe we'll catch up for a coffee.
Yeah, well, let me know, because my parents live just north of Birmingham, so I do occasionally pass by. I'm from Staffordshire originally.
Oh well, all right. Well, I'll be back in Blighty soon.
Excellent. Well, enjoy.
Right.
Good to see you, Michael. Hopefully catch up
Thank
again
you
soon.
all. Have a great day.
Bye.
Thanks,
Bye.
everyone. Bye.